Influenza A virus is the subtype of Influenza A virus that was the most common cause of human influenza in 2009. It is an Orthomyxovirus that contains the glycoproteins Haemagglutinin and Neuraminidase. For this reason, they are described as H1N1, H1N2 etc. depending on the type of H or N antigens they express. Haemagglutinin causes red blood cells to clump together and binds the virus to the infected cell. Neuraminidase are a type of glycoside hydrolase enzyme which help to move the virus particles through the infected cell and assist in budding from the host cells. Some strains of H1N1 are endemic in humans and cause a small fraction of all influenza-like illness and a small fraction of all seasonal influenza. H1N1 strains caused a small percentage of all human flu infections in 2004–2005. Other strains of H1N1 are endemic in pigs and in birds. In June 2009, the World Health Organization declared the new strain of swine origin H1N1 as a pandemic. This strain is often called swine flu by the public media. This novel virus spread worldwide and had caused about 17,000 deaths by the start of 2010. On August 10, 2010, the World Health Organization declared the H1N1 influenza pandemic over, saying worldwide flu activity had returned to typical seasonal patterns. Swine influenza. Swine influenza is a respiratory disease that occurs in pigs that is caused by the Influenza A virus. Influenza viruses that are normally found in swine are known as swine influenza viruses. The known SIV strains include Influenza C and the subtypes of Influenza A known as H1N1, H1N2, H3N1, H3N2 and H2N3. Pigs can also become infected with the H4N6 and H9N2 subtypes. Swine influenza virus is common throughout pig populations worldwide. Transmission of the virus from pigs to humans is not common and does not always lead to human influenza, often resulting only in the production of antibodies in the blood. If transmission does cause human influenza, it is called zoonotic swine flu or a variant virus. People with regular exposure to pigs are at increased risk of swine flu infection. The meat of an infected animal poses no risk of infection when properly cooked. Pigs experimentally infected with a strain of swine flu that caused the human pandemic of 2009-10 showed clinical signs of flu within 4 days, and the virus spread to other uninfected pigs housed with the infected ones. During the mid-20th century, identification of influenza subtypes became possible, allowing accurate diagnosis of transmission to humans. Since then, only 50 such transmissions have been confirmed. These strains of swine flu rarely pass from human to human. Symptoms of zoonotic swine flu in humans are similar to those of influenza and of influenza-like illness in general, namely chills, fever, sore throat, muscle pains, severe headache, coughing, weakness, and general discomfort. The recommended time of isolation is about 5 days. Notable incidents: Spanish flu. The Spanish flu, also known as La Gripe, La Gripe Aspar plus or minus Ola, or La Pesadilla, was an unusually severe and deadly strain of avian influenza, a viral infectious disease, that killed some 50 to 100 million people worldwide over about a year in 1918 and 1919. It is thought to be one of the deadliest pandemics in human history. The 1918 flu caused an unusual number of deaths, possibly due to it causing a cytokine storm in the body. The Spanish flu virus infected lung cells, leading to overstimulation of the immune system via release of cytokines into the lung tissue. This leads to extensive leukocyte migration towards the lungs, causing destruction of lung tissue and secretion of liquid into the organ. This makes it difficult for the patient to breathe. In contrast to other pandemics, which mostly kill the old and the very young, the 1918 pandemic killed unusual numbers of young adults, which may have been due to their healthy immune systems mounting a too strong and damaging response to the infection. The term Spanish flu was coined because Spain was at the time the only European country where the press were printing reports of the outbreak, which had killed thousands in the armies fighting World War I. Other countries suppressed the news in order to protect morale. Fort Dix outbreak. In 1976, A novel swine influenza A caused severe respiratory illness in 13 soldiers with one death at Fort Dix, New Jersey. The virus was detected only from January 19 to February 9 and did not spread beyond Fort Dix. Retrospective serologic testing subsequently demonstrated that up to 230 soldiers had been infected with the novel virus, which was an H1N1 strain. The cause of the outbreak is still unknown and no exposure to pigs was identified. Russian flu. The 1977–1978 Russian flu epidemic was caused by strain influenza A 90/77. It infected mostly children and young adults under 23 because a similar strain was prevalent in 1947–57, causing most adults to have substantial immunity. Because of a striking similarity in the viral RNA of both strains A1 which is unlikely to appear in nature due to antigenic drift it was speculated that the later outbreak was due to a laboratory incident in Russia or northern China, though this was denied by scientists in those countries. The virus was included in the 1978–1979 influenza vaccine. See also 1889–1890 flu pandemic. For the earlier Russian flu pandemic caused either by H3N8 or H2N2, 2009 H1N1 pandemic. In the 2009 flu pandemic, the virus isolated from patients in the United States was found to be made up of genetic elements from 4 different flu viruses: North American swine influenza, North American avian influenza, human influenza, and swine influenza virus typically found in Asia and Europe. An unusually mongrelized mix of genetic sequences. This new strain appears to be a result of reassortment of human influenza and swine influenza viruses, in all 4 different strains of subtype H1N1. Preliminary genetic characterization found that the Haemagglutinin gene was similar to that of swine flu viruses present in U.S. pigs since 1999, but the neuraminidase and matrix protein genes resembled versions present in European swine flu isolates. The 6 genes from American swine flu are themselves mixtures of swine flu, bird flu, and human flu viruses. While viruses with this genetic makeup had not previously been found to be circulating in humans or pigs, there is no formal national surveillance system to determine what viruses are circulating in pigs in the U.S. In April 2009, an outbreak of influenza-like illness occurred in the United States and then in Mexico. The CDC reported 7 cases of novel H1N1 influenza. By April 24 it became clear that the outbreak of ILI in Mexico and the confirmed cases of novel influenza A in the southwest U.S. were related, and WHO issued a health advisory on the outbreak of influenza-like illness in the United States and Mexico. The disease then spread very rapidly, with the number of confirmed cases rising to 2,099 by May 7, despite aggressive measures taken by the Mexican government to curb the spread of the disease. On June 11, 2009, the WHO declared an H1N1 pandemic, moving the alert level to Phase 6, marking the first global pandemic since the 1968 Hong Kong flu. On October 25, 2009, U.S. President Barack Obama officially declared H1N1 a national emergency despite President Obama's concern. A Fairleigh Dickinson University Public Mind poll found in October 2009 that an overwhelming majority of New Jerseyans were not very worried or not at all worried about contracting the H1N1 flu virus. However, the President-unregistered trademark S declaration caused many U.S. employers to take actions to help stem the spread of the swine flu and to accommodate employees and / or workflow which may be impacted by an outbreak. A study conducted in coordination with the University of Michigan Health Services scheduled for publication in the December 2009 American Journal of Röntgenology warning that H1N1 flu can cause pulmonary embolism, surmised as a leading cause of death in this current pandemic. The study authors suggest physician evaluation via contrast-enhanced CT scans for the presence of pulmonary emboli when caring for patients diagnosed with respiratory complications from a severe case of the H1N1 flu. The March 21, 2010 worldwide update, by the UN's World Health Organization, states that 213 countries and overseas territories communities have reported laboratory-confirmed cases of pandemic influenza H1N1 2009, including at least 16,931 deaths. As of May 30, 2010, worldwide update by World Health Organization, WHO, more than 214 countries and overseas territories or communities have reported laboratory-confirmed cases of pandemic influenza H1N1 2009, including over 18,138 deaths. The research team of Andrew Miller MD showed pregnant patients are at increased risk. It has been suggested that pregnant women and certain populations such as Native North Americans have a greater likelihood of developing a T helper type 2 response to H1N1 influenza, which may be responsible for the systemic inflammatory response syndrome that causes pulmonary edema and death. Recent developments: on April 26, 2011, an H1N1 pandemic preparedness alert was issued by the World Health Organization for the Americas. In May 2013, 17 people died during an H1N1 outbreak in Venezuela, and a further 250 were infected. As of early January 2014, Texas health officials have confirmed at least 33 H1N1 deaths and widespread outbreak during the 2013-2014 flu season, while 21 more deaths have been reported across the U.S. 9 people have been reported dead from an outbreak in several Canadian cities, and Mexico reports outbreaks resulting in at least 1 death. Spanish health authorities have confirmed 35 H1N1 cases in the Aragon region, 18 of whom are in intensive care. On March 17, 2014, 3 cases were confirmed with a possible 4th awaiting results occurring at the Centre for Addiction and Mental Health in Toronto, Ontario, Canada. According to the U.S. Geological Survey and the CDC, northern sea otters, off the coast of Washington state, are infected with the H1N1 flu virus and may be a newly identified animal host of influenza viruses. In pregnancy, pregnant women who contract the H1N1 infection are at a greater risk of developing complications because of hormonal changes, physical changes and changes to their immune system to accommodate the growing fetus. For this reason, the Center for Disease Control and Prevention recommends that those who are pregnant to get vaccinated to prevent the influenza virus. The vaccination should not be taken if you have had a severe allergic reaction to the influenza vaccination. Additionally, those who are moderately to severely ill, with or without a fever, should wait until they recover before taking the vaccination. Pregnant women become infected with the influenza are advised to contact their doctor immediately. Influenza can be treated using antiviral medication, which are available by prescription. Oseltamivir and zanamivir are 2 neuraminidase inhibitors currently recommended. It has been shown that they work best when taken within 2 days of becoming sick. Since October 1, 2008, the CDC has tested 1,146 seasonal influenza A viruses for resistance against oseltamivir and zanamivir. It was found that 99.6% of the samples were resistant to oseltamivir while none were resistant to zanamivir. In 853 samples of 2009 influenza A virus, only 4% showed resistance to oseltamivir, while none of 376 samples showed resistance to zanamivir. A study conducted in Japan during the 2009 H1N1 pandemic concluded that infants exposed to either oseltamivir or zanamivir had no short-term adverse effects. Both amantadine and ramantadine have been found to be teratogenic and embryotoxic when given at high doses in animal studies. Additional images. Notes. External links: European Commissioner Euro Public Health EU Coordination on Pandemic 2009. Health EU Portal EU work to prepare a global response to influenza A, H1N1. Influenza Research Database of influenza genomic sequences and related information. Centers for Disease Control and Prevention H1N1 Flu. Consultant Magazine H1N1 Center, Pandemic Influenza, A Guide to Recent Institute of Medicine Studies and Workshops. A collection of research papers and summaries of workshops by the Institute of Medicine on major policy issues related to pandemic influenza and other infectious disease threats. H1N1 Flu, 2009, Hearings before the Committee on Homeland Security and Governmental Affairs, United States Senate, of the 111th Congress, first session, April 29, 2009, coordinating the federal response. September 21, 2009, Protecting Our Community, field hearing in Hartford, Connecticut. October 21, 2009, Monitoring the National Unregistered Trademark S response. November 17, 2009, Getting the Vaccine to Where It Is Most Needed. Non-technical, Shriev, J., Why Revive a Deadly Flu Virus? New York Times. A six-page human interest story on the recreation of the deadly 1918 H1N1 flu virus, 1918 flu virus's secrets revealed. BBC News. September 28, 2006. Results from analyzing a recreated strain. Neumer, some information on TB and the 1918 flu. Data from Neumer, Garin M. The 1918 influenza epidemic's effects on sex differentials and mortality in the United States. Popol Dev Rev 26, 565 €81 doi, 10.1111 per Joule 1728-4457.2000.00565x. PMCA 2,740,912. PMIDA 19,530,360. Oral history by 1918 pandemic survivor, technical, Ludwig S., Horsteiner, Kaliter E.F., Scale T6 C. Recent influenza A infections of pigs and turkeys in Northern Europe. Virology 202, 281 €6 doi, 10.1006/0. 1994.1344. PMIDA 8,009,840. Centers for Disease Control. Influenza A, H1N1, associated with mild illness in a nursing home. Maine. MMWR Morb. Mortal. WKLY. Rep 36, 57 €9. PMIDA 3,100,930. Lederberg J. H1N1 influenza as Lazarus, genomic resurrection from the tomb of an unknown. Proc. Natal. Accord. Sci. USA 98, 2115 €6 doi, 10.1073/FNAS 05100078. PMCA 33,382. PMIDA 11,226,198. H1N1 registry.